I'm Professor Kumar and you are learning at Galaxy Online Learning. Today we are doing an important chapter, animal tissues. Every chapter looks important, but this is really important because it is a fundamental chapter. You have been studying from right 8, 9, 10th also, and has more weightage in NEET and MHT CET biology exam also. It tests your fundamentals. Biology depends on the tissues. That is why. Okay, our whole physiology is linked to tissues. We will see animal tissues. First, just from your lower class, what is a cell? Cell is structural and functional. Your body cannot be made without cells. Your body cannot function without cells. Actually, cell is got protoplasm and is surrounded by plasma membrane. These are minimum things required in any cell. Then, protoplasm has a nucleus and it has got organelles. Mitochondria, Golgi are those. Okay, so we have given examples. All these are also there. Endoplasmic reticulum, mitochondria, Golgi bodies, centrioles, lysosome, etc. Many more. Each organelle, each of these does a particular job. There is division of labor inside a cell. Cells can be studied as two groups, somatic cells and germ cells. Soma means body. They are body cells. Your entire body is made up of it. Okay? They do body activities, digestion, enzyme, secretion. Germ cells are called sex cells. They only do reproduction. Like they are present in the testes and ovaries. Okay? Have a look at how epithelium from your mouth, ciliated from the trachea, from the stomach, columnar, muscles from the intestine, striped muscles from your arms, from the heart, okay, cartilage cells. See the different type of cells present in a tissue. Okay, we'll go to the next slide. Now, what is a tissue? Tissue, first, not a single cell, group of cells. Should have look same, structure should be same, and should come from the same source. What are the same source? Okay, remember three germ layers, endoderm, mesoderm and ectoderm. Okay, but they may have a little mixed variety, then they are called compound tissue. Because an organ can't be made of pure one tissue. So, organs are formed from tissues and organs together form organ system. What is histology? The branch of science where you study tissues under a microscope is called histology. Microscopy is important. Okay, my friends? What are the four types of tissue? Don't write from a 9th standard plant tissues. Animal tissues are epithelium, connective tissue, muscles and nervous tissue. Don't write plant material. Also known as, for your magna column and MCQs, covering tissues. They protect. Connective are supporting tissues. They bind. They are contractile tissues, movement and locomotion. They are conducting tissue, conduction of nerve impulses. Very brief. We are going in details. Have a look. Animal tissues are of four types: epithelial, connective, muscular, nervous. Epithelium is squamous, cuboidal, columnar, ciliated, glandular. Of course, there is sensory, germinal, reproductive, etc. We are doing main types. Connective tissue is areolar, adipose, skeletal, and fluid. Areolar is in tendon and ligaments, very important for our 11th standard. And skeletal is in bone and cartilage. Fluid is in blood and lymph, but we study this in the circulation chapter. We do not study it here. Not to repeat, so we study it there. Okay, so this is the types of tissues we are going to study in this chapter, each short note. Have a look of the four. Why there are four tissues? Type of cells, arrangement of cells, secretion of cells, space between the cells and the surface below the cell. Type of cell, they are all similar, squamous, cuboidal, columnar. Connective, different type of cells are seen, histiocyte, mast cell, adipose cell. We will be seeing in detail with diagrams. Muscular, all in the muscle are same type. Nervous, all are neurons. How are they arranged? Very tight, very loose, very tight, and okay. Their secretion does not enter the tissue, it comes out of the tissue. Secretion remains in the tissue. It helps the connective tissue to grow. Does not enter into the tissue, and it is inside the tissue, adrenal, etc. No space between the cells. Large space, no space, moderate space. Here it is stuck on a basement membrane, stuck, not stuck. Okay, we will see epithelial tissue now. First type of tissue in the body, epithelial tissue. What is epithelial tissue? It covers outermost surface and lines the inner cavity. So it is a covering and it is a lining of your body. One or more layers, tightly packed, no much gap. Stuck on basement membrane, stuck, okay? Membrane name was given, we don't want to change, non-living. Don't forget, they are not cells, that's a secretion, like a cement, and the tile is fixed on it. These epidermis will come from ectoderm or mesoderm lining the coelom, or endoderm, your midgut, intestine and stomach region. We will be studying later in embryology. Cells don't have blood supply between them. Okay, below is the blood in the connective tissue. Okay, they can divide very fast, they can repair very fast, and they keep coming off. Every time we have a bath, all the dead cells keep coming off. And they may be smooth, they may have hairs, ciliae, or in the intestine they may have comb-like microvilli. Okay. Now, what are the types of epithelial tissue, my friends? Simple, compound, one layer, more than one layer. Simple squamous, simple cuboidal. We don't write simple every time now. Squamous, cuboidal, columnar, ciliated, non-ciliated, two types of columnar. Glandular, sensory, germinal. Here, stratified and transitional. Okay, we are going to see all these now. First, simple epithelia. Squamous, one by one. See how it looks like your pav you're getting from the market. Okay. It is found in peritoneum. Peritoneum is a lining like sellotape which is sticking the organs to your body. Okay. And lining of the blood vessel so that blood doesn't leak out. It's present on the surface of the skin. When you drink hot coffee, see it comes out, white membrane will come out. Cells are polygonal in shape, thin, flat. Okay, nucleus is in the center. They appear as tiles. So also called pavement, footpath, pavement epithelium. What does it do? It filters and lets material to go into your skin. Oxygen, nutrients can cross. Bacteria and foreign particles can't go. It will prevent that. Okay? So short note, 2 marks. How does it look in the microscope? See, this is how it looks in the microscope. Those are drawings and these are actual photographs. You see, this is you can see a blood vessel which is cut and see squamous, how thin the layer is. Okay? Nuclei can be seen stained dark purple. So it gives you an idea of shape. Second type, cuboidal. Now these are like your small cakes. Found in the thyroid, kidney. They are cube shape. Cubic cube. Nucleus is in the center. Round or spherical. They absorb and secrete. 2 main important work they do. Okay, so have a look at the diagram. How does it look actually? Have a look at the down. I'll show you the actual photograph. See how cuboidal looks from a side. Actual photographs. Okay? So you can have a look. See? Now epithelium, squamous, see cuboidal. Everything can be seen nicely. Let's go to the third type. Simple columnar. It lines your stomach and intestine and other organs. Cells are tall, they are like pillars, like chalk. It is broader, it is little narrower at the top, okay? And there is a basement membrane on which it is stuck. Nucleus is oval or ellipse and it's present basal, below the half line. Okay? See how it looks actually. See the nucleus is towards the base. See how tightly packed they are. Gaps is relatively less. You can see. Beautiful. So these are actual versus the drawing, artist drawing. Ciliated columnar. You see, these are cilia. This is columnar, this is cilia. It is found in your respiratory tract, nose, esophagus, everywhere. Okay? And fallopian tube, so that the egg can go into the uterus. Okay? Cells are tall and pillar-like. This is free end anterior, this is stuck on a basement membrane. Okay? And cilia come out, protoplasmic extension, and there are basal granules from which they come out. Okay? Then nucleus is round or oval, mostly oval, and always towards the lower half. Okay? Cilia vibrate and they can push, as you have been learning, your nose, nasal passage, all the mucus and particles are trapped in the mucus. See actual photograph, I will show you. So you can have a look, how does it look actually. Have a look. See, basement membrane can be seen, the cement-like substance. Nucleus is towards the base. Cilia you can see, hair-like, small hair-like particles are coming out from this end. Okay? Cilia. Simple glandular epithelium. They make enzyme, hormone, sweat, oil, all secretion, milk, whatever you talk of. They are carried into ducts, tubes, ducts, and this is called a gland because it is a specialized structure. Two type of glands are seen: endocrine gland and exocrine. Endocrine and exocrine we are going to have a look now. Okay, then below the slide. Endocrine secrete into the blood. They don't have tubes. They are made and put to the blood. Pituitary, thyroid, parathyroid, ovary, testes, adrenal, Langerhans islets. Exocrine have ducts or tubes. They are made somewhere and carried. They are responsible for body secretion. Tear, saliva, acid in the stomach, intestinal juice, all are made in this manner. Exo outside. Now have a look how the exocrine, this is not there in your book, so you can have a look. Various shapes of exocrine gland which are seen. And I have written example. This is more important for NEET and MHT CET. See, respiration, stomach, upper portion, lower portion, okay, sweat glands, see, and duodenum, intestinal, mammary gland, milk is produced, pancreas, pancreatic juice. So they acquire various shape. Either they are test tubes or they are alveoli, which is like bulbs. And they have seen ducts or tubes through which secretion comes out. Everybody has a mouth through which it will come out. Okay? Sensory, again, your book is avoiding it much. It is modified columnar and it's got these nerve fibers which come out. Hairs are present, sensory structures are present. Okay? And they can see outside and inside. Okay? When you sleep, your sense organs are still working. Okay? You can find out it's cold. I need a blanket. It's hot. Switch on the fan. Okay? I can feel that I have to urinate. All the feelings are there by receptors. Found in the nose, your sense organs, you know. Nose, ear, eyes. 5 sense organs. This is found in the sense organs. What is compound epithelium? Stratified in skin, dead layer on the top, which comes out. Keratin is the membrane, is the protein, and basement membrane, and the living layer is down. This divides and comes up, divides and keeps pushing, and the top flat layers keep shedding off. Transitional is in between. When I am forming an organ, the sole, the palm, so I will have transitional epithelium in between. So that it occupies space. Okay, you're going to change shape as you grow older. Okay, your book does not deal again with this detail. Okay. Now, epithelium and epithelium are joined by junctions. How do they communicate? The blood is down in the connective tissue. Except sensory, others don't have nerve supply. How does one cell tell another what to do? Okay, are we ready? There are tight junctions, adherent junctions, desmosome, gap junction, focal adhesion and hemidesmosome. Okay, tight junction seals neighboring cells, will not come apart. Adherence joins actin bundle between cells. Okay, actin bundles will join with. We are going to see all details. Huh? This is just a summary chart. Desmosome, as you can see, has got filaments, like how you put iron rods in the floor of cement and make RCC, the floor joints, or POP, you put those threads, nylon threads. Okay, gap junction, so gaps are there, so I throw from your window you pick up. Okay, focal adhesion, they just joined by like a shoelace, you just tie in between. Okay, hemidesmosomes tie the cell to the lower part. So the cell has to stick and not come. If I pinch your skin, it doesn't come out. So it will tie it down. You see? Beautifully explained. 6 types. We are going to study what is there in our portion in details. Okay? Let's see what the textbook shows. Diagram of the textbook is impossible to follow. Please have a look to understand better. Tight junctions. They come very close to each other. Like I stitched 2 things together. Okay? So you can see who discovered for your NEET, we don't need for theory, Farquhar and Paley, 2 plasma membranes seal and join like sellotapes. We have 2 proteins, claudins or claudins, positive, negative, binding. Internal organs and cavities and skin does not come out. Digestive, dark cavities. Why? Prevent leakage. Imagine if your urine leaked inside your body. Then blood-brain barrier. We don't want all chemicals to go to the brain. Our brain will fail with the poison. Okay? They maintain polarity. Which direction to which direction? Can you enter or can you exit? You can't do both. They have got polarity. Have a look. Okay? How they look like. See? You're locally tight junctions. And there are special proteins you saw. And they are like rivet. Tight junctions. This is from one cell. This is of the other cell. Okay? Then adherence junction. Here it's like we put solution, rubber gum, glue and then Fevicol and then we stick some mica to the. So this is how we are doing. You can see microfilaments are there. Microfilaments. So they look like plaque, dark spots, heart muscle. Because I have to contract also. If I put tight, it will tear. The bridge always has gap. Railway tracks have to have gaps. Okay? So heart, layers of the body organs because organs keep moving. Digestive tract keeps peristalsis. Okay? And signals can pass through. Transcription, how much RNA, what to do, all information can go through. See how I see. These are like those links between the bridges ka spans. Okay? As I told you, our diagram in the books are harder, so it's very difficult to understand. So have a look. Desmosomes. In a cell, how would they be looking? And how are they actually? Cell 1, cell 2, and we have got tick button type. One has got surface and it will go and link inside, like Velcro. Okay, desmosome. So plasma membrane has cadherins. There is a gap between and there is a plasma membrane too. And then there are intermediate filaments. Okay, you don't need such diagram detail, but since the photos are so badly there in your book, you can't understand, we have made it clearer. Who discovered? Porter. Desmo means bond. Where is it found? Skin, lining of cavity, because needs a lot of flexibility. Cadherins are present. These cadherins join 2 cells together like Velcro. Okay, and they can stretch. You can always pull Velcro. It will slightly open, it will not leave. Become tighter and tighter. When cells are transformed, when they become something else, they are aging or they change, then these things go and the dead cell can be thrown off. Okay? Gap junctions. See how beautiful? We have left gaps. Okay, but gaps of control. Huh? Gap doesn't mean a channel where you can just run away. Okay, these are like policemen is there on both sides. So there's a complex. You can see how 7 proteins are there. 6 proteins are connexins. Okay, connexins, and they look like donuts. And there are channels. It can open and close the channel. Important in heart. The whole heart has to beat at the same time. We can't have one auricle contracted, the other did not contract fully. Okay, one relaxed, other didn't. Both didn't relax. Can't have all that. Gap junction is important. Calcium runs through these gap junctions. Okay, so iron and small molecules and chemical messages can run. So they have left the channel for that purpose. Gap junctions. Okay. Then hemidecimal. These are like an anchor of a ship. Okay, ship. Such a big ship also needs a small anchor. So the extra cell, they go and link the cell to the bottom frame so that the cell won't move or come out and won't destroy itself. Okay, and there is signaling allowed. So homeostasis, tissue can remain nice. So this is the cell, this is the outside, and these are the links. And in details, you will see. So nicely explained to you. Plectin family. Okay. Then we will come to connective tissue. By the way, the junctions are only one picture in your textbook. Okay. Connective tissue. They come from mesoderm, not ecto and endo. They are most abundant. Most of the tissue of the body, maximum tissue of the body is connective tissue. There are many cells. Okay, there is a matrix background, and then this background allows diffusion. Then there is cells. And different tissues, more cell, more matrix, less matrix. They have blood supply, highly vascular. Except blood, other tissues have fibers. Blood does not have fibers. Okay? And fibers are of collagen or elastin, which we are going to study in details. What are connective tissues? Connective tissue proper. The job is only to connect. Areolar, adipose, white fibrous, yellow elastic, reticular. Give support, cartilage, bone. Cartilage, hyaline, fibrous, calcified. Fibrous is white or elastic. Different type of movement plus support. Bone, pure support for movement. Compact bone, spongy bone. And vascular tissue is studying in the circulation chapter. So in our chapter we will not cover this detail, so that we don't repeat this. Picture showing you all the connective tissues as much as we could. Okay, take your arm. If you cut a piece of arm, adipose is there, cartilage is there at the end of the bone is there, blood vessel, blood is there, loose connective tissue is there in the packing, and fibrous connective tissue is there, which is joining the bone to bone like cellotape. See, all tissues can be seen like this. Together, we study one by one. Okay? So this is the diagram which shows you location of connective tissues. Let's come to areolar connective tissue. It is connective tissue proper. Below skin, below muscles, below bones. What does it do? It is loose connective tissue. Only connects. Skin is joined to the muscles. Skin does not come off from the muscles. Okay? Nerve vessels are present surrounding. They don't move, they don't come off. Then peritoneum, like sellotape. When I run, my stomach does not run. My heart does not run outside. They are sellotaped with this. And muscle fibers are tied in a bundle. We can see bundles because of this. Structure, it has got transparent matrix. It's got fibers, it has got cells. What are the fibers? White, yellow. Of course, third reticular is there, which you study for NEET and CD. But white, unbranched, wavy, and in bundles. Okay, white in bundles, wavy. Okay, they are made of collagen. They are like nylon rope. Yellow are slender. Okay, they are branched. They are flexible, and they are like rubber bands. Okay, now see how two different objects. One is like nylon ropes, thick, will take a lot of weight and all, and second will let you stretch, move, vibrate, and it will like elastic, like rubber bands. Okay? Okay. These are the two type of fibers. Then we see the diagram. See how beautiful diagram, and see your textbook diagram. Okay, see the various type of cells, how does it look loose. Okay, now you can see fibroblast, adipocyte or fat cells which are also present in the skin, macrophage, mesenchymal cell are type 4 type of cell C. Fiber, you can see collagen fibers in bundles like a cable, and elastic fiber like thin wire but branched, and reticular fibers, as you will see, very fine ones. You have to stain with silver and see under electron microscope. They are like jelly, net, holding the tissue. Tissue does not tear. Your teacher pulled your ear so many times, your nose so many times. It does not tear because of the reticular fibers. And the remaining is matrix. Secretion is not empty. It's just like a jelly or custard. Okay? Now, cells are fibroblasts. These fibroblasts are star shaped. They make fiber, fibroblast. So the matrix is secreted by them. Macrophage, they are like amoeba. Okay, and their job is amoeba. Phagocytosis, capture and destroy soldier of the body. Mast cells produce chemicals. Okay, big nucleus, and of course I showed you adipocytes with fats inside them. So 4 type of cells can be seen, and 4 different functions. Break the matrix and fiber, capture all the germs, okay, make the chemicals, and give the store food and give reserve food for energy. Okay. Adipose is found below the skin, subcutaneous, covers the heart, covers the blood vessel, and in the bone it is yellow bone marrow, not red bone marrow. It is a food reserve, prevents heat loss, absorbs shock, and produces blood corpuscles. Okay. Structure. Okay. We have got adipocytes which store fats. So sometimes adipose, from areolar we make adipose, we throw all the structures we keep only adipose cells. Now adipose cells are of 2 types, white and brown as we call it. Now when there are cell organelles like mitochondria, Golgi, appear dense under the microscope, gives a brown coloration. When you are born, you have brown adipose bone. But as soon as the cell becomes specialized, stores only fat, it throws out the cell organelles except the nucleus. Okay? Only white fat remains and becomes white adipose. So the adipocytes give 2 colors, and see how the diagram, the 2 is brown, that's white, how different they look. Same tissue. Once it has become white, that fat will not go from your body, it is specialized. That is why if you don't maintain, your fat will come back. Okay? And brown is mitochondria and all, can always reduce itself. Okay? Next. White fibrous tissue is present as tendons, and we will see later the yellow as ligaments. Tendons, you know, your bones are joined, okay, and to the muscles. Okay, now suppose I take 200 kilo and I am lifting. Now the bone, muscle is pulling the bone, okay, and the tape between the muscle and the bone gone forever. That's why sports people, fast bowler, you see, and Sachin Tendulkar, now he says, I get pain. Stop. Fast bowler, shoulder goes. Okay? Because, and weakest point of your body, okay, and hamstring, you know, when you jump or leap and you get a hamstring, you get the pain you get. So, these are difficult to repair. They take a lot of time to repair. Okay, because there is cartilage inside. Okay, between muscle and bone. So, surgeries also are not very simple. Now, sports medicine has come up. You see those blue tapes they apply, cricketers and sports persons. That's to localize and prevent the cartilage from getting more damaged. Certain movements are restricted. Let's come to yellow elastic. Yellow elastic is bone to bone, and they are called ligaments. Okay? Now you know bone to bone, when I'm open, close, open, close, movement. So unless there is elasticity, the bone will come out. It's not some screw, metal plate or something inside. So this is that important thing. Okay? And of course, when it goes, you're gone. That's why when the back breaks, finish. It's very difficult to repair a yellow. So you can see, those people who put artificial knees and they cut the cartilage as they were doing in earlier time, chop, chop and put metal, not successful. Today they keep all the ligaments in place. That is why very highly successful. Not still 100% because fine cartilage you can't see. Okay? But today we keep the ligaments, we don't cut the ligaments. Let's see the difference because distribution between. Your book is not doing justice. That is white, this is yellow fibers. Here continuous rows, they are scattered. Here skeleton, muscle to bone, here bone to bone. Tough, inelastic, strong, elastic. Opposite properties, so easy to remember. Okay, diagram not expected here. So then we come to cartilage. When you are born, you have more bones because you are cartilage in nature more. And when the cartilage becomes totally bones, bones fuse, cartilage joins, your bones reduces number. Okay? So cartilage is covered with white fibrous tissue like cellulite. Amazon packing, how it comes. It is called perichondrium. Perichondrium is not cartilage, it's the covering of the cartilage. Cover of the book is not the book. Perichondrium has inside matrix called chondrin, and toward the ends are cells called chondroblast. Chondrin, chondroblast means young cells. The matrix is made by the cells. It is a fluid film, jelly-like fluid, and the cell is inside an empty place. That's called a lacuna. Lacuna, empty space, gap in between cell, and there is fluid in that gap. Okay? When it grows, because of the gap, the cell can grow, can divide, can form more, and nutrition comes. Gas exchange occurs because of the fluid in that hole. Have a look. Have a look. Hole, cell are there, hole is there, there is liquid in the hole, and there is matrix. Okay? Now you can have a look. So materials can exchange, blood vessel can exchange throughout, each cell to cell, diffusion. Now cartilage is of 4 types. Hyaline, fibrous, elastic, and calcified. Okay, 4 type of this cartilage. By changing the fiber, the cell, basic structure is same. We change the composition, the proportions, and we make types. Okay. First, hyaline cartilage. This is like that bouncing ball you buy. When you throw it, comes up very high, transparent, bluish. Okay. It is the weakest end of the long bones. Every bone at the end has got this, so that when they rub against each other, no friction is coming. Compressible, we can compress. Elastic, and of course covered with cellulite, it doesn't come out. Okay, and very fine collagen fibers are present in it. Flexibility, support, reduce friction, and shock absorber. When you jump, you know how much the shock the bones are taking. Okay, so diagram shows hyaline cartilage ka localization. Fibrous cartilage. Now we have got a peculiar problem. My head is on top, the whole weight is coming here, and when I turn, I am supposed to look. I am supposed to tell you, put some books on your head and tell you, oh, my neck is paining. The whole day body is carrying the neck and the head, it is not paining. These duster-like clothes are there between the vertebra. So I can turn, I can bend limited, and I do not get pain, I do not feel friction. These are fibrous cartilage. Pubic symphysis between my leg joints. If this were not there, I would never be able to jump or sit cross-legged. A woman would never be able to give birth because the girdle has to become big, birth canal has to form. Very important. Now when this damages because of smoking, alcohol and bad posture, effect is permanent. So what do? We just take a platinum plate and put with screws. So you will be like, you cannot repair. Very difficult. Okay, tough like duster cloth. So, elastic cartilage, very nice as I told you, your ear, your nose, nothing. See the pig, the dog, every time the cow is touching the black nose to the ground, stones, cactus, thorns, nothing happens. Okay, because it is elastic cartilage. They can move the skin, the ear, they can move, the tail it can move. It's because of this cartilage. Chondrocytes are few and we have got more of the yellow fibers. It gives you shape and support. And lastly, we have got calcium. When we put too much calcium into it, looks like bone, functions like bone, but is not bone, can grow. So salts is deposited, cartilage is hard, it loses the flexibility. In old age, when joints lose the mobility, all becomes cartilage. That is why we get arthritis, pain, can't get up soon. Okay? So that is why this kind of calcified cartilage. Let's have a look. Distribution between blue, white, yellow. Few fibers, lot of white fibers, lot of yellow fibers. Flexible, firm, very flexible. On top, no sellotape. Sellotape is there, sellotape is not there, sellotape is there. They're found in rib, trachea, bronchus, larynx. Found in intervertebral disc and pubic symphysis. This is found in the external ear and nose tape and all these organs. So made your life very simple. When you have a look, your text becomes simple. Bone, everything is important. Bone protein is called osine. Remember, cartilage was chondrite. It is made up of hydroxylapatite, Ca10, PO4 and OH twice. Learn this for the exam. Okay, and we will see details of the bone. Have a look. So when we have bone, inside we have these circles. One circle looks like this, and in a circle, one of this looks like this. The cell is here, and the cell can connect to other cells. He is in an empty place, lacuna, and there is fluid, brown. So each is joint. That's why bones, when they break, they pain, because you cut canalicula have so many. But in humans, because the circles are so many, when I break, they join very fast, because they are just little. That's why any bone of your body, when it breaks, within 3 months it is done. Actually, in 3 weeks it is done. Bone is covered with periosteum outside and osteum inside, so calcium cannot leak. Second, it's got layers. We saw circles, lamellae. And they have empty place called lacuna. And in lacuna are osteoblasts, the blue color cells. Okay? They join by canaliculi, fibers to the neighbors. Okay? Now this system is called Hevesian system after William Harvey, who found that there are canals which connect each other. Okay, and blood vessels and nerves go through it. So bone is not a dead tissue. Lymph vessel, blood vessel, nerve vessel enter the bones through these Hevesian thing. And the connections which are the central are Hevesian and the lateral are Workman's canal, named after the scientist. That is, the bone grows longitudinally. Okay. Function of the bones, I did not, we were studying in the 8th standard. Protect, support, give shape, store, potassium, calcium, phosphorus. Okay. 2 types of bones are there. Spongy bones and compact bone. Spongy at the end of the bone, because of friction and all, where cartilage is present, it is spongy. The middle part of the bone is strong, can take weight. It's like tire and the frame of the car. Okay? So, here osine has got tuberculae, branches with gaps, and they contain bone marrow which is soft. Here, there are no gaps, it has got yellow marrow and Hevesian system. Okay, there is no Hevesian system in spongy bone yet, because we can't drill holes in soft, they will collapse. They give strength but not much weight. These fat, weight, everything. You can hit a person with a bone and his bone will break. Okay? Muscular tissue. It comes from mesoderm, not ectoendo. Again. And 40% of your weight is this. It has got fibers. The cells are long. They are called fibers. They can contract because they have got myosin and actin proteins. Okay? And we didn't know. Today we know all cells are the same. We saw muscle cytoplasm. We could not see it. It was like solid. So So sarcoplasm is a cytoplasm of muscle. The fibers are myofibrils, and there are nerve fibers inside and blood vessels which supply the muscles. Okay. We will have a look at muscles. 3 types of muscles are found in animals which are studied in lower class: striated, non-striated, and cardiac. So you can see heart, cardiac, in your locomotory organ skeletal, in your internal organs, smooth muscles. And the shapes are also seen. We are studying in details. Striated muscles. They are mainly skeletal, so attached to the bones, head, trunk, limb. Common name skeletal muscles. They show dark and light bands, and they are parallel to each other and they are in a bundle, like how you have cold drink or straws or a pencil box. Okay? And those pencils or straw are like myofibrils, and the gap between the pencil is the sarcoplasm. Okay? And the bands are light and dark. So I band and A band. I for isotropic, A for anisotropic. Light can pass, light cannot pass through it. And the gap between the dark and light is the Z-line sarcomere, between 2 adjacent ones. Okay? And between 2 light is called an H-line or Henson's line, named after the cycle. We have seen the diagram. Do not worry. Have a look now. Okay? So there is a cover on the top, there is cytoplasm inside, and there are these pencil-like objects, myofibrils, inside. Now you can see Z-line can be seen on them. Okay? I band can be seen, A band can be seen, where those white structures there. Okay? So this is how a muscle actually. Nucleus is pushed to the side. Peripheral. Now, how does one unit look? Between 2 Z-line is a sarcomere. Sarcomere is a structural and functional unit of a muscle. Okay, please have a look. So, middle M-line, Z-line, 2 neighbors, between 2 Z-line, sarcomere. If I keep repeating this, I will get a complete myofibril. What are these lines? Have a look. There are these channels and there are these windows, sliding windows or umbrella. Okay? Now these move between the sliders. So I can make it big, I can make it small. Won't come out. The spikes don't come out of umbrella, window does not come out of the channels. Volume does not increase, mass does not increase. Only sliding occurs. Okay? So I have sliders and I have things which moving inside. And because light now passes, so it will hit this, it will hit this, it will hit this, it will hit it. So I will see dark lines. Here, it will hit, little lesser. So I see like this. Okay? So where I get more line, I will get like this. So light coming out, there is no color inside. Okay? So have a look. And this is how it appears under the microscope. See the striations, how the sarcomeres can be so clearly. See, unit, and nucleus is on the sides. Okay. Smooth muscles. They are in your internal organs, stomach, intestine, etc. They are called viscera. Viscera is inside, visceral muscles. They are involuntary, you cannot control much. They are elongated or spindle shaped with tapering ends. Spindle, you know, you're used in your seen in school also. Your mother makes laces. Spindle shape. Okay? And they are bound by sarcolemma, like cellotape, together. And the 2 type of nerve supplies come in. Sympathetic, positive, for example, and parasympathetic, negative, for example. Okay? They are involuntary, you cannot control. They are slow, but long time. Stomach can keep working, but you can't make it work 10 days continuous eating. Okay? And contraction is slower, faster. How the body requires peristalsis, movement of food, how you push out toothpaste, it pushes the food inside. All this occurs with this muscle. Look now how the cells come together. All nerve supplies are stuck on top, and then we put serotonin on this. See? Now ciliary muscle of the eye. This is it. Stomach. This is it. Okay? See how they are under a microscope, how they look. See, so clear spindle shaped, you can see the pointed ends. Nucleus in the center. And myofibrils are those dark lines you see longitudinal. So I will show you actual versus the drawing so that you are easy for your practical exam and understanding. Okay. Cardiac muscles are striped also, but like smooth, they are like more spindlish and they are network connected to each other because the whole heart has to beat at the same time. Okay, so it is found in the myocardium, middle layer of the muscles of the heart, which help it to beat or contract. Okay, now that's why it says in between. It's got properties of both, but it will never get tired. It follows its own pace rhythm, contract, relax. Okay, and third, most important, intercalated discs are there. Cytoplasm from one current. If I put my head down, everything will come down, my upper heart will become empty, nothing like it. I lie down, top heart will become empty, nothing like it. Cytoplasm and others can't go. Intercalated discs which are like a toll gate between the two. You just can't cross like that. Okay. Now, heart beats itself. We know that. And then the whole thing switches on and off. Okay. So, we have now made a pacemaker, battery with 2 wires which you put in the heart. If your heart is not beating or beating is very poor, and it will give sparks, and the entire heart will be pacemaker. Human heart is myogenic. The muscles produce the impulse. The brain does not produce the impulse. If I remove your heart and keep it outside, it will beat for another 40 minutes. Okay? And those which are neurogenic are controlled by nervous system, and if I cut that heart, it will not beat unless when the brain has been separated. That is why we can transplant heart. It can fly. It can be brought and put into another human being. Okay? Have a look at the cardiac muscle, how beautiful they look under the microscope. See the branches? Very casual branch. See? It's 3D, huh? That's why now you'll tell me, why this branch comes from down up? It's 3D. We are showing you 2D pictures. Okay? It's branching top, bottom, left, right, everywhere. It's like a mass. Okay? But the difference can be easily seen. What is the difference between the three? Voluntary, involuntary, cardiac. Body wall, limbs, etc. Hollow viscera, iris and skin. Heart only and vena cava. Here mitochondria, glycogen is solid. Here very few. Here solid. Here T system distribution channels. Trabeculae. Here no trabeculae. Here trabeculae is there. No disc separates the two cells. No disc separates the two cells. This separates the branches. Can control, cannot control, cannot control. Unbranched, unbranched, branched. Blood supply, large blood supply, less blood supply in capillaries. Does not enter the muscle. Heart never gives blood to itself. Blood vessel from top brings the blood. Myofibrils are big. Here cannot see much. Here we see them very faint. Quick, very fast. Stomach slow. And here powerful. See, distribution trees. So easy to remember. Then we come to nervous tissue. It is coming from ectoderm, outside layer, germ layer. Then neurons. Impulse can give, impulse can conduct, and impulse can understand. Excitable and conductible. No other tissue can. Okay? Two important properties must remember. Then it's got neurons and some nerve cells, supportive cells called neuroglia. Short from glia. Let's have a look how they look. See, under a microscope, this is how it looks. And these are neuroglia. The sides. Okay? We draw in a very star-like manner, but this is how they look. And see how the axon goes behind. Yeah. Structure. Outside cell membrane, neurolemma. It has got cyton cell on the top. Cyton is the cell on the top. Cyton. And rope-like axon. 2 parts. Okay. Now, cyton has got granules inside. It's got cell organelles. and these granules are RNA. Because Nissal discovered them, we named it after him, Nissal's granule. Today we know they are RNA. Okay? And these which come out are dendrons. Okay? So, axon and dendrons are extensions and cyton is the cell. Have a look how to draw diagram. Okay? Now, don't forget, message comes from dendron into the cell and goes from axon, goes to dendron of another cell, goes to axon, like that. There is polarity. You can't do whatever you like. Dendrons receive, they take the catch. The captain tells what to do and the axon will throw. A catcher can't throw, a thrower can't catch, and a captain will decide what to do with it. Okay, so this is why it is important. If there is fat, insulation is called myelinated. If this insulation is not there, okay, it is called non-medullated, non-myelinated. So two types of cells are seen, which we will see. Dendrites are thin, small ones. They carry impulse towards the cell body. They are very few. Axon is single and long. It takes the impulse away, and inside axon, if I cut the axon, I have exoplasm inside. Okay? And it also contains mitochondria, which you can't see and all. Okay? And it has got Schwann cells on top, cellotape on top. Schwann cells on all. These are Schwann cells. Okay? They contain myelin. So if the myelin is there, it is myelinated. If the myelin is not there, it is non-myelinated. Why do we have this insulation and no insulation? Myelinating a cell is a very expensive proposition. When you are born, only important nerves which have come from the mother are myelinated. As you grow, you feed yourself, you myelinate other nerves. Myelination stops after a certain age, because the body starts aging. So all nerves never get myelinated. Only the important ones are myelinated. So sometimes when you want to get up, you suddenly fall down. You see, message did not conduct properly because it was a non-myelinated which conducted. But my heart can't decide. I will not beat. Okay. Now you will tell me, why did the body in evolution keep it? Non-myelinated are important. If I want to pass information very fast, okay. Now I have two choices. I have to show my neighbor current has come, okay. Either I will pass the wire, he will pull the wire, put a bulb and see, or I will touch the wire to a wet wall and tell him to touch the wall. Which you think is the fastest? Of course, the wet wall. The body leaks outside the body. This is the axon. It leaks it outside, it travels the liquid and again enters. I am drawing too close. So see how fast the electric impulse jumped. Saltatory conduction. Important for your survival. Suppose a car comes, immediately go back. Car is coming, please turn back by the time the car will go over you. So this is very important for survival and reflex action. So body has left these nodes of Ranvier also. Okay, and stop myelinating after some time. Okay, you can live with it. Okay, so these are the things in it. Chemicals made, the endings, where the nerve fiber ends, we draw it like a synaptic plate. And here we will have these chemicals, neurosecretory material, acetylcholine and adrenaline. These chemicals will go in the surrounding space, will enter a dendron and then go to another cyton. So message crosses by chemicals. The wire does not go into the next cell. So once the current is come, it will throw chemical will go into the other cell, again produce the current. Okay? So that will show you why saltatory conduction is more faster. Okay? Sometimes leakages are better. When the paper leaks, even you are happy and I am most happy. How a nerve is formed. Okay? Now there are several bundles. Fasciculi. Okay? Endoneurium, perineurium. See the cellotape around, cellotape around everybody. Cable, red, green, blue and black, aluminum wire, and still one more cable on top of it. That is how your nerves are protected. So that they can function well, and when we move, bang, sleep, jump, they don't break. Cables don't break. You can see how beautifully they are arranged. Okay? Now neurons, depending on the structure, are unipolar, bipolar, multipolar. Upon the work they do, they are sensory or motor, and according to, as I told you, fat, they contain, they may be modulated with myelin or without myelin, non-modulated. Bipolar comes from two ends, unipolar comes from one end, multipolar comes from all the ends. So where is the signal coming from? Different will have different. They have different functions. It's not just better, that is what body has to use how it requires. Now eyes, rods and cones, we require from one side only. I can't have something coming out from my eye, something going into my eye. These are made for functioning of the body. Okay. Now myelinated, non-myelinated from your 8th standard, fat is there, fat is not there. Okay. And I told you how leakage occurs and how the signal jumps, can go through or can jump across and re-enter somewhere else. Okay. Slow, fast. But here everybody will get a message. Function not good. When you get a shock, you don't feel very happy. Your life has been saved. Okay. Yes, my friends. So thank you very much. Two things I must tell you. Please register so that you can have access to all these diagrams and question answers, board questions, MCQs, label the diagrams, draw the diagram, all you will get. They are ready. And second, and you can ask difficulties when you go through those question answers. When you see this slide, see them very slowly. Keep a notebook and a pen. Please take down the charts, notes, points. They're beautiful. Very difficult to study. We are not boasting. When you try, you yourself will thank us. And they will remain in your mind for a longer time. Remember, you need it after 12 for entrance exam also. And if you're doing engineering also, it will help you study biology very easily without putting too much stress. Secondly, since I'm putting it on YouTube so that you don't have to download, bandwidth, problems and all, and you can watch it 100 of times, you can slow, you can pause, you can make it bigger, smaller. So please register on that bell icon, subscribe, so that you know when I'm putting up my next video. Individually for me it's very difficult to first put these lectures on record and then upload them, label them, playlist. It takes time. You can access it as soon as I put, if you have subscribed to the bell icon. Okay, so two things. Register as fast as you can. You can meet personally, bring difficulties, take assignments, take question papers, and join the NEET portion also, or CD portion. And second, and CD NEET lectures are still different from these, with difficult topics included, words included, past paper questions included. Theory and NEET MCQs are slightly different. Okay, so kindly do so and contact us. Any suggestions, any changes, anything we can help you, we are there. Okay, thank you.